All right, everyone, welcome back for the final video lecture for the biodiversity unit. And this video will be on selecting desirable traits. So essentially, when you're selecting desirable traits, you are choosing the outcome that you want. And there are many methods that this could be done through, either through artificial selection or biotechnology. And there are a couple categories in biotechnology that we will be going over, including cloning, artificial insemination, in vitro fertilization, and genetic engineering. So with artificial selection, it's essentially the process of selecting and breeding individuals with desirable traits to produce offspring that have these desired traits. And this is not a new practice. This is a practice that has been going on for tens of thousands of years. So one of the examples of artificial selection is all these vegetables here come from a relative wild mustard. And so the desired traits that are seen in these vegetables, they were selected from the wild mustard and crossbred to produce the vegetables we have today. Same thing goes for dogs. The different dogs that we have today, they all have a common ancestor of the gray wolf. And in different parts of the world, dogs were bred to have certain desired characteristics. Now, biotechnology. So biotechnology is essentially a high-tech way of speeding up the artificial selection process. So with artificial selection, you have to wait a couple of generations to see the traits that you have that are desired. And biotechnology is used by farmers, breeders, and scientists. So the first biotechnology we're going to be talking about is cloning. So cloning is essentially you're creating the exact replica of the parent, and the DNA is going to be exactly the same. So if you're cloning, let's say, some plants that you really enjoy the traits of those plants, instead of crossbreeding them, you could take a cutting of a leaf from that plant, place it in some water, let it develop roots, and then place it in the soil. However, there is limitations with that because there are only so many leaves that can be typically cut off a plant, and so it limits the amount of clones you could create. A way they have found a way to create more clones for plants is taking the cells from a plant, letting those cells develop in nutrients in either a petri dish or water, and then planting those cells. So you get definitely a lot more plants from that process. And a similar thing happens with cloning for animals. You're essentially removing the DNA from the egg and placing the DNA that you want within a fertilized egg, and then placing that egg in a surrogate mother to then produce the cloned animal. Artificial insemination artificial insemination is essentially artificially implanting the male sperm inside of the female. And so the sperm will be harvested from the male, and then this will be implanted into the female. We often see these on ranches or in zoos in captive breeding programs. The captive breeding programs are often running to help animals that have a small population regain that population. They're assisting essentially in creating another generation. And it's also popular in the beef industry. The beef industry relies on artificial reproductive technologies. Artificially reproductive technologies so that the desired traits in the meat that the ranchers are looking for are for sure going to then end up in the next generation. In vitro fertilization, you're taking the male and female gametes and that have been selected, and then you are having the fertilization happen in a petri dish. So that's the main difference between in vitro fertilization and artificial insemination. You're selecting the two gametes, and the fertilization happens in a petri dish, whereas in artificial insemination, the sperm is just being inserted into the female. With in vitro fertilization, you are able to determine what the gender is ahead of time, and then select which fertilized eggs you want to implant into the surrogate mother. And then there's genetic engineering, which is directly altering the DNA of an organism. An example of this being bacteria is used to produce insulin for humans. And when you use genetic engineering and you're selecting the specific DNA that you want to have in the next generation of the organism. And you can see in this image here, with conventional breeding, you end up getting all the genetics in the next generation. But with genetic engineering, you are selecting specific traits that you want, combining them together to have only those traits be passed on going forward with the next generations and generations going forward as well too. Now, there are a lot of risks that come with playing around with genetics. Some of those risks include reduced variation. And when you have small variation, this makes you more susceptible to diseases. So if all the cattle on a ranch are related, are brother and sister because they all came from the same bull, then if there was a disease that those genes were susceptible to, that entire ranch would then be wiped out. Same thing goes for plants as well too. A whole field could be wiped out because all of the plants are related and have the same genetics. Same thing for cloning, your same DNA, so you would have the same unfortunate fate. There's also health issues with cloning as well too. We've seen unsuccessful pregnancies, birth defects, and deaths among clones. And so the technology behind cloning isn't really perfected yet. And then as well with crossbreeding. So we see artificially selected plants can still sometimes crossbreed with the original old wild relatives. An example being cauliflower still being able to then breed with its wild mustard relative. But where the danger comes in here, say you have canola that has been genetically engineered to be resistant to herbicide. That canola could then crossbreed with the weeds that the herbicide is trying to spray and get rid of. And when that crossbreeding happens, you end up with weeds that are now resistant to the herbicide as well too. And so it's creating new genetics or new generations that were unplanned for. And that's essentially it for topic 4 and your biodiversity unit. I believe you will now have some unit review questions that you can do to help you guys study for your test. And as always, please let us know if you have any questions about the information from this video. And thank you for watching.